All right, hello everybody. This is your video check-in for mutations. So let's go ahead and talk a little bit about what happens when the sequence of DNA gets changed somehow. Okay, we refer to this as a mutation. So any change in the DNA is referred to as a mutation. So we see here on in this picture, right, we have some flowers. A mutation in these garden moss roses has caused some of the flowers to grow orange rather than yellow, right? So a change in the DNA caused different proteins to be made, and therefore different pigment is being produced, right? So mutations can actually be fatal, meaning that it kills the organism. It could be negative, meaning that it harms the organism. Okay, it could be benign, has no effect, doesn't help it or hurt it, or mutations are sometimes beneficial to the organism. So let's look at an example of a fatal mutation. An example of that would be albinos in corn plants, so albinism in corn plants. Here we see one of these corn plants is completely white. The plant does not make chlorophyll pigments and therefore is not able to do photosynthesis. So it was able to sprout because there was energy in the seed that allowed it to get started, but this plant is going to die because it's not going to be able to do photosynthesis and make sugar for its energy source. An example of a negative mutation would be a mutation in hemoglobin, the gene for hemoglobin, which causes the hemoglobin protein to misfold. And when the hemoglobin misfolds, that actually changes the shape of the red blood cells. And we can see on the left we have normal red blood cells. On the right we have what are called sickle-celled. And these sickle-celled red blood cells are unable to pick up as much oxygen. So this actually does not kill the organism or the person that has this disease, but means they can't carry as much oxygen to their cells, which means that they're going to become very, very tired, and just doing something like walking up some stairs can make them out of breath. So this is definitely a negative mutation. Benign mutations. Benign means doesn't help, doesn't harm. So we see this flower has a mutation in some of its petal genes. So the genes on the right have a mutation in them, whereas the genes on the left lack that mutation. And this isn't going to hurt the flower, and it's also not going to help the flower either. Do you think that these mutations would get passed on to the offspring? So that's a question we want to be thinking about, and we'll come back to that in a minute. Right, beneficial mutations. All right, so if you didn't notice, there are two moths in the picture. There's the black moth on the left, and then there's the speckled moth on the right. So a mutation in the moth on the right caused a change to the pigment genes, and the moth on the right appears speckled. So we want to think about, why do we consider that a beneficial mutation? And would we always consider this to be a beneficial mutation? So I'd like you to go ahead and pause for a second and write down why this is a beneficial mutation, and if this would always be considered a beneficial mutation. And we'll follow up on that in class. So go ahead, pause, write your response. All right, welcome back. So how do mutations happen? All right, so let's take a look at this whippet. And whippets are little racing dogs, but this whippet has a genetic disorder called double muscle that results in a very muscular appearance. So do you think this is a beneficial mutation? Why or why not? Okay, and if you said, yeah, it's a beneficial mutation, actually, this is a negative mutation. Even though she has twice as much muscle, she doesn't have any more circulatory system or a stronger heart. So actually all those muscles require lots and lots of oxygen, and so when she tries to run or do things, she gets tired extra fast because she can't get enough oxygen out to all of those muscles that she has. So this is actually considered a negative mutation. So when you think mutation and you think about beneficial mutation, doesn't necessarily mean bigger or stronger. Okay, it means more adapted to that organism's environment. So when did this mutation happen? Did it happen during mitosis or meiosis? Right. So if you said, this mutation must have happened during meiosis, you were right. How do we know that? Because it affects her entire body, which means that this mutation happened is in all of her cells. When we think back to the flower, it was only in some of the cells, which means that was a mutation in mitosis, and therefore isn't only a group of cells are being affected. So when we think about mutations and how mutations get passed on from parent to offspring, only mutations that happen in the gametes in the sex cells are going to get passed on. Okay? And the reason for that is because those mutations in the gametes, right, are going to go into all of the body cells. So a mutation that maybe you have in an arm cell or something like that, a cell of the arm, right? That mutation would not get passed on to your kids, right? It would only be the mutations that are in the gametes because that's where new individuals come from. So how do mutations happen? So we have things called an error in replication. So we've talked about DNA replication and how DNA copies itself. Okay, and then we also have mutations called chromosomal alterations. We're going to talk about those as well. So when we think about what can cause mutations, things out in the environment called mutagens. Mutagens are anything that can cause DNA to change. So replication errors. So what is this? This happens when the DNA is changed. A base pair gets changed in some way. So you can think about these as something like a spelling error. So when the DNA is getting copied and you're making new, the DNA is copying, right, making two sets for two new cells, sometimes the wrong nucleotide can be inserted or a nucleotide can be eliminated or one added in. Okay, when this happens, right, you change the DNA, which then will change what type of RNA gets made and then what protein gets made, right? So a change in the DNA can affect the RNA, which in turn affects what protein gets made. And if you change the structure of a protein, you probably are going to change its shape and its function. So let's talk about sources of error of replication. So point mutation, okay, is a substitution of a single base pair. So one base pair gets changed. So here I have a sentence. This is not DNA, but it's like DNA in that, hey, we have 3 letters in each word, just like in the language of DNA. 3 letters makes up a codon. And so here I have my original sentence that reads, cat ate the rat. And I see I have a mutation here. The T was changed to a P. Now it reads, cat ate the rat. So the one mutation, that point mutation, changed one word in my sentence. All of the other codons were unaffected. An example of that would be like we saw with sickle cell anemia, where we had just one amino acid changed, and it changed the whole structure of the protein. Frameshift mutation. A single base pair lost or a single base pair is added. Now, okay, my T is still there, but a P was inserted between the A and the T. And that moved everything over one, right? So we say that the reading frame is changed in a frame shift. So everything what we call downstream after the mutation is affected. So my original sentence read, cat ate the rat. It now reads, cat apt, eth, t. Right? So everything after the mutation was affected because all the codons were changed since we shifted everything down one. So we say that all codons downstream of the mutation are affected. Usually point mutations are not as significant as a frame shift. A frame shift can actually completely change the protein product. Right? An example of that would be Tay-Sachs disease in humans. So we now want to talk about other types of mutations called chromosomal alterations. So there's actually several types of chromosomal alterations, one being deletion, inversion, translocation, non-disjunction. Okay, so let's talk about deletion first. So a deletion is when part of a chromosome gets left out or deleted. Right? So here we have our little image showing part of this chromosome got deleted. Okay. Other types of chromosomal mutations include inversion. This is when a piece of a chromosome breaks off and reattaches in the opposite order. Translocation is where a piece of one chromosome breaks off and attaches to a non-homologous chromosome. So maybe a piece of chromosome, I don't know, chromosome number 3 breaks off and it reattaches to chromosome number 12. That would be an example of a translocation. Right? And also want to talk about duplication. This is when a piece of a chromosome is repeated, right? So it gets duplicated, and now we have a longer chromosome, right? So that's an example of a duplication, right? And then finally, non-disjunction. So when we think about the chromosomes getting pulled apart during the process that happened during cell division, usually the sister chromatids get pulled apart to opposite ends of the cell. But in a non-disjunction mutation, one cell is going to receive a duplicated chromosome, whereas the other cell receives no chromosome at all. What this means is that you end up with an extra chromosome in one cell. Okay? Having an extra chromosome changes the chromosome number. Right? An example of a syndrome caused by non-disjunction is Down syndrome, right? Where a person has an extra chromosome number 21, right? So if we look at this person's DNA, their chromosomes, this is a person with Down syndrome, and we notice they have 2 of every chromosome, except for chromosome number 21, they have 3. So that extra genetic material actually causes them to have some genetic abnormalities. All right. So, want to talk just briefly about what things in the environment can cause changes in DNA, cause mutations. Okay, those things in the environment are referred to as mutagens. So, an outside agent that causes mutations. Some examples include things like ultraviolet rays, right? So a reason when you go to the beach you should wear sunblock is because the high energy rays in the sun can actually cause mutations in your DNA. Radiation, right? So things that are radioactive can cause changes in your DNA. Some chemicals are able to cause changes in your DNA, and even some biological things like viruses cause changes in your DNA. An example of that would be the human papillomavirus, HPV, and it is a virus that actually causes mutations in the DNA and is known to cause cancer. So the good news is there's a vaccine for this virus. So if you get vaccinated and I think it's 2 shots you have to get over the course of a year or two. That means that you'll have resistance against this virus, and if you come in contact with it, you won't get the virus, which reduces your chances of getting cancer, right? So it's always a good idea to try to prevent disease. So that's just an example of a virus that causes mutations in DNA.